Recording about an example of a receptor and in its signal transduction pathway and cellular response and so forth. And in the same way that we talked about muscle differentiation to help us understand the genes that need to be activated for muscles to form and the transcription factors that regulate those, we're going to learn about this story of receptor biology and the normal healing of a wound that is on our skin. And so it's something that happens underneath a scab every time we get cut. And the skin heals, right? And so when the scab falls off, there's pink new skin there, new skin cells. And so we needed to stimulate that healing under the scab. So that's what this is about. So it's a practical example. And as molecular biologists, we don't exactly care about the reason. Instead, we are really interested in the molecular signaling pathways underlying that. But just like with the clay model videos, I think it's really helpful to put it in a context. So we use this example over and over again. But just like with the muscle genes, we're not interested in just this receptor. There are lots of receptors and receptor stories that we want to talk about. So our example is going to be getting a cut in your skin, and the main players that we're going to talk about is a factor that's going to bind to the receptor, and it has a good name. It's called platelet-derived growth factor. And the receptor is going to be on our skin cells, and it's going to be what responds to this PDGF, which is released from platelets at the site of a wound, and this is going to stimulate the receptor to activate a signal transduction pathway. It's going to feature a lot of protein phosphorylation, and there's a lot of talk about that in receptor biology and in our course, so we want to just make sure we're solid on that. And then this cellular response is the skin cells go through the cell cycle to grow and heal the wound. So that's the layout of it. Here's a Wikipedia page on protein kinases. The chemical activity of a kinase is, well, it's an enzyme that catalyzes a chemical reaction. The chemical reaction is to transfer a phosphate group from ATP, typically ATP, to a target. And the target could be a protein or another molecule that gets phosphorylated, and we'll meet both kinds. There are a lot of enzymes that do that job in our body, and they activate or inactivate target molecules, and then that chemical reaction must be reversible, and so there's another group of enzymes that reverse that. Those enzymes we'll talk more about. They're called phosphatases. So a kinase phosphorylates, and a phosphatase removes the phosphate. And there are lots of these. There are 500 or 600 protein kinases in our body, and each has a gene. So 500 or 600 protein kinase genes that we inherit from our parents, and a pretty big part of the total human genes, maybe about 2% of total human genes, and at least. And of these 500 or 600, one of the molecules that we'll learn a lot about is our receptor, and our receptor is called the PDGF receptor, and it's one of these 500 kinases. So it's in that group, and of those, it's in a category of receptors that all have that same quality, that they function by phosphorylating targets when they're activated. So the PDGF receptor is a receptor that is a kinase, and it phosphorylates tyrosine amino acids on target proteins. So it's called a tyrosine kinase, and then it's a receptor. So this is called a receptor tyrosine kinase, and we have 58 of those in humans. And we'll talk about some other ones as well. For example, the insulin receptor is a receptor tyrosine kinase. And so these receptors are really important. They really often are involved in growth, And so that's one reason we'll be especially focusing on them, because our book emphasizes them, because our book leans toward talking about cancer biology. And we'll see today in this video our first example of how mutations in the gene for these kinases can sometimes contribute to cancer, like that. This is from IntroBio and from the chapter that's about cell signaling in every IntroBio textbook. And this shows us a vision of what's going to be happening here. There's the cell membrane, and in our case, that's going to be a skin cell, epidermal cell, like that. Here comes a signaling molecule, and it's the PDGF. So we must be at the site of a wound, and a scab is forming because another set of molecules that get released from platelets. Let's just draw a platelet up here for the moment. Platelets famously release clotting factors that help clot the blood at the site of a wound, and then also helps form a scab. And they also release PDGF, and that's good because we want to simulate healing under the wound. And PDGF binds to the PDGF receptor, and then a signal transduction pathway takes place, and a cell response takes place. And in our example, the cell response is going to be focused on the cell cycle and increasing the rate of cell proliferation. So it's going to be, this is a growth factor, PDGF, platelet-derived growth factor, like that, and that's the PDGF receptor. Both these are proteins, so they must have genes. This protein must be made in this cell, so the PDGF gene must be, PDGF receptor gene must be active in this cell. This cell has the PDGF gene, but it's pretty important that it's not made there. It's made elsewhere, and it's made where platelets are formed, and we'll talk about that right now. So this is what we're talking about, the epidermis of our skin, body covering, and that's what's going to get injured during a wound, and then we're going to have platelets release clotting factors, form a scab, and also release PDGF under the scab to stimulate the cell cycle. Platelets come from the bone marrow. And so here we are in the bone marrow, and most of you are probably familiar that continuously throughout our life, we have bone marrow stem cells that are producing new blood cells all the time. So when we donate blood or we bleed a lot, we get stimulation of new red blood cells. And even if we don't do either of those things, our blood cells are constantly being replaced. And that's also true of our immune system, our circulating immune system cells, like white blood cells are involved in, are being formed like that. And then these platelets are formed too. And I have a picture of that idea. So the platelets are formed in the bone marrow by one of these cells that's constantly being replenished, but is not sent out into the blood. And the cell is called a megakaryocyte, and it's in our bone marrow all the time, and we always have fresh ones. And bone marrow cells, called megakaryocytes, pinch off of the cell membrane little packets, and these little packets are platelets. And a platelet is just a little portion of a megakaryocyte that's pinched off. It does have a phospholipid bilayer. It's the original cell membrane of the megakaryocyte cell, and those are what come out into the circulatory system. And we have lots of them, and they don't last very long because they aren't exactly alive. They don't have a nucleus. I don't think they have mitochondria. They're just little packets like that. And it's unusual, but that's how platelets are formed. Lots of times when people donate blood, they'll ask them just for their platelets, because their platelets are something that are really important post-surgery, when you're healing, when you need to clot blood, you need a lot of platelets in there, or a wound, a car accident victim, or after a surgery, and so forth. And these platelets, I think, only last for about a week or something like that. So they're constantly being replenished. These megakaryocytes themselves are being replenished, but they last for a while, and they're just constantly pinching these off. Now, let's just look at this. Here's where PDGF gets made. So again, all the cells in our body have the PDGF gene, but the PDGF protein is only expressed here. So we got a PDGF gene there, it's active, and it's transcribing and then getting translated by ribosomes, and we just have PDGF in the cytoplasm. It's not outside of the cell, it's in the cytoplasm. And that means anytime a platelet forms, a little bit gets pinched off of the cell, and that includes lots of PDGF. And so here's a circulating platelet, and it has lots of PDGF inside of it, as well as clotting factors. Clotting factors are made here at this megakaryocyte as well, because they're in these things. And we can think of a platelet as like a little suitcase that's just circulating the blood, and it has really important things in it, but you only want to use them when you need them, and you want them ready, circulating continuously, because you don't know where and when you might get cut and start bleeding and need to release some clotting factors and some PDGF. And of course, there are other stories of the platelets and what they're releasing and what their, what their role is in all kinds of stuff in our body. But the key thing is we're really talking about two cell types: the epidermis or skin, and then the megakaryocyte, which is part of our blood cells, but actually remains in the bone marrow and pinches off platelets continuously from there, which circulate in our blood. Okay, so that's what we're looking at. And then we're looking at the cell cycle, and we're specifically looking at skin cells. Now, here's the thing about skin cells. It's a good example because skin cells are constantly dividing in our life. And you're familiar with the idea that our dead skin cells rub off all the time, and we're constantly replenishing them. So skin cells are slowly going through the cell cycle. And if we put on this map of the cell cycle, if we put, say, 100 skin cells, they would be scattered at different points along the cell cycle because they're constantly dividing, but it's regulated. And the most important place where they are is most of them are just stopped here at this, what's called the G1 to S transition, and we have a chapter on the cell cycle coming up after our receptor chapter, so we'll learn a lot about this, but just a kind of a superficial terminology so we can get that. A more common term for this is called start, because it's really typically where the cell cycle starts. The cells are stopped here, and then they receive a signal, like from a growth factor, like from PDGF, through the PDGF receptor signal transduction pathway, and they move through start. S phase is really kind of a robotic, phase. It just kind of goes through that, copying the DNA. So that's where the DNA gets synthesized like that. We copy all the chromosomes. And then in our bodies, G2 is also usually just kind of a thing that the cell gets through, and then the cell divides. G1 is where the cell spends most of its time, most of its living is here, where gene expression is occurring and stuff like that, and it's just doing its thing. Okay? So like muscle cells are in G1, and that's such a common thing that there's actually a special part of the cell cycle people refer to as G0, and we'll use all that terminology, and hopefully you learned a bunch of it in your first year intro bio course. So I'm going to draw some circles to show you, there are lots of skin cells at start, waiting. 100 skin cells, maybe 90 of them, like that. Just made that number up, but the majority of them are here. And then the rest are just scattered throughout the cell cycle. And they're just scattered there because they got the opportunity to go through start. They sort of got a green light, and they're just going to continue through the cell cycle until they get around to start and pause again. So out of 100 cells, maybe 85 or 90 of them are here, and the other 10 or 15 are just scattered across the cell cycle. And that's because the skin cells are constantly dividing at a slow rate, like that. And so it might even be higher. It's probably higher than 90. And because they're dividing slowly, and what that means is they sit here a long time. It's not that the cells going through the cell cycle are going through it slowly. It's that they're paused for a long time, and then they're released, and they go through it and come back to start and stop again. And that's why it's called start, is because they're really waiting there. They're in G1, so they're doing their regular living. They're just doing their skin cell living, expressing genes and stuff like that, and waiting for a signal to go through the cell cycle. What we're going to find is that this is the normal skin cells constantly dividing. Under a scab, we want to increase the cell cycle rate, And so it's not that the cells go through the cell cycle faster, it's the cells under a scab get released from start more frequently, and it's because of PDGF. So that's going to be our cellular response to PDGF, is that the skin cells are going to go through the start more frequently. We're going to stimulate that start step like that, okay? There's a rate limiting step, why these guys are paused here, and the PDGF through the PDGF receptor and the signal transduction pathway is going to increase that rate limiting step. And that's going to involve some changing gene expression like that. Okay, great. So here's just an example of a test that's very often done by a doctor, and where they'll take some cells and they'll test it. And so if we just took a little patch of cells, of your normal cells, and we looked at it under a microscope, if we counted 100 cells, 100 just normal skin cells, we would find some of them are in mitosis. And it's maybe like 2%, something like that. Just 2 or 3 out of 100 are in mitosis. Now, why mitosis? Because, because that's the only time we can see things happening under a microscope. That's when the chromosomes condense and the cell divides. And so what they're doing here is they're saying, yeah, there's 100 cells, and we just want to know how often they're going through the cell cycle. So we're just going to look where we can see these, and if we see 2% here, well, there'd be a couple percent here and a couple percent there like that. There'd be like 92% at start. So counting the mitotic index is an indirect way of counting what's happening at start. When PDGF stimulates the cell cycle, cells get to go through start more frequently, and that's going to increase the mitotic index. And doctors have been doing that for years and years to study health. So anytime a patient comes in with a lump or a bump or an odd mole, and the doctor takes a biopsy of that tissue, they will take some of the cells and prep them on a microscope slide, and a pathologist in the clinic lab will count 100 cells and determine the mitotic index, the percentage of cells that are in mitosis. And that's why I've been saying 100 cells, just to make the math easy, right? So if it's 100 cells, the mitotic index would be 2. Now, when they do that under a scab, the mitotic index is higher. And it's higher because the skin cells have been stimulated to go through the cell cycle. And people say they stimulated to go through the cell cycle faster. They should say more frequently, like that, okay? And so we can measure that, and like I said, on a biopsy, doctors are doing that for unusual cells, and cells of concern, like an odd-looking mole or something like that, or a lump or a bump, and they measure the mitotic index. And the doctor will come back and say to the patient, well, I'm not too concerned that that tissue, that bump, is growing slowly. And what they mean is the mitotic index is low, or the mitotic index might be high. And they're like, there's no reason for it to be high because of a wound. It wasn't a wound. There must be some other thing going on. The mitotic index might be high, and even higher than a normal healing skin cell, might be high, and they say, I'm worried it's growing fast. Over here, they'll say it's growing slowly. I think it's benign. Over here, they say it's growing fast. I'm concerned it might be malignant. Now, that's where the patient's got concerns over a lump or a bump, but just their normal skin cells behavior provides the context for the doctor to understand, like over here, if we have a biopsy of a mole, well, how does that mole mitotic index compare to the relatively slow but not zero rate of normal healthy skin growth, and how does it compare when the cell's growing pretty fast, right? So we have a nice comparison to do that biopsy like that and measure that and come up with feedback for the patient. So here's just an example of the mitotic index. The mitotic index is predictive for survival in dog, so pet dog, cutaneous mast cell tumors, like that. And so they're just saying, we measure the mitotic index, and they've been doing it for many, many years, certainly, I don't know, like 100 years, because they can see it under a microscope. It's a pretty simple assay. And so they're measuring it in a certain type of dog cancer, and here's what I highlighted. It said the median survival time for dogs with a mitotic index less than 5% was significantly longer than for those with a mitotic index above 5%. So that we'll see, as we talk more about cancer, there's a lot of other ways, additional ways now, that doctors understand what's happening in a cell that is in biopsy tissue because of concern about a lump or a bump or an oddball and stuff like that, right? Or a polyp, right? When somebody gets a endoscopy, and anywhere in our body. And so this is just a good example here when dogs had a relatively lower mitotic index, below 5. They lived for a few years. Above it, they lived for a couple of months, like that. Okay, and there's a lot of data on that in humans as well. And so same idea here, just shown again. And a lower mitotic index in an odd mole might suggest to the doctor that it's benign. A high mitotic index suggests it might be malignant, and it's just measured that same way. Again, lots of additional things and more all the time, and some of which we'll learn about are being used to assay and understand what's happening in cells. Okay, so we're going to talk about a certain type of cancer, and it's a type of skin cancer, and it is very rare. You don't know anybody that's had this kind of skin cancer. It's just a very rare type of skin cancer, and doctors diagnose it in a specific way, and they've diagnosed it for years in that specific way. Okay, and the specific way is, again, this cancer, dermatoma, fibrosarcoma, protuberans, is associated with a chromosomal translocation. And a chromosomal translocation is a kind of mutation where a chromosome gets broken by accident, and when that happens, the chromosome can be put back together, can get joined back together. However, sometimes, so it's a type of mutation that can get repaired. Sometimes 2 chromosomes are broken at the same time, and by accident, the cell will join this to that chromosome piece, and it'll join this chromosome piece to that chromosome. The chromosomes in humans are numbered based on their length. So we have chromosome 1, the longest, 2, 3. And so they're saying that there's a chromosomal translocation, and they can see it when they do a karyotype under a microscope. So when they take that tumor tissue and do an additional assay where they look closely at the cells that are in mitosis, they can actually see a problem with 2 of the chromosomes. The chromosomes are number 17 and number 22. And they see that 17 and 22 have been broken and have gone through this chromosomal translocation. They can test that during a biopsy. So in this case, this patient had a biopsy, and they said, oh, the mitotic index is high, we're concerned, and in looking at those mitotic cells, we can see that you have undergone this specific chromosomal translocation, and so that patients that have that are said to have this type of skin cancer. Okay, and so they've been knowing that for years. I'll just guess that this patient might have lived in the 1950s, and they were able to see that there. Now, they didn't have any idea why this was a problem, but more recently, because of the physical nature of DNA, scientists have been able to analyze what happens at this chromosomal translocation. And what happens is something that we are going to be very familiar with when we draw it out, and that is 2 genes get damaged. And I don't know what chromosomes they're on, but let's say, just say this is maybe the chromosome 17 gene, and this is a chromosome 22 gene. And in modern times, we can look at the DNA along that chromosome, and those chromosomes that undergo that translocation that lead to that kind of cancer, they go through a specific translocation by accident where this chromosome breaks here and this chromosome breaks here. Those chromosomal breaks happen by accident, a random mutation, at these positions, and when the two chromosomes are repaired by accident, they join, and I'll just draw this like it is a broken chromosome, like I drew before. This broken chromosome is accidentally sealed up to this broken chromosome, which is part of the longer chromosome in my original drawing. And so, now we'll say that was the shaded chromosome, and instead of repairing it and correctly joining that broken piece back to this one, an accident is made. And so we're going to join, when we repair, we're going to join this part of this gene to this part of that gene. And it's a random mutation, an accidental mutation, and it happens, of course, at a specific location. And that specific location is what causes the problem, because it turns out that this is a transcription unit. I guess I should put that label on there. So, right, we're joining the transcription unit of one gene with the cis regulatory region of another gene. And we know that we could do that on purpose in a test tube. We can join the regulatory region of the myogenic gene to the transcription unit of GFP, and we could get muscle in a transgenic mouse that will glow because the GFP is made under control of the myogenic regulatory region. So this is very much like that laboratory experiment, except it happens in nature, in humans, by accident. And of course, it's very unfortunate. And what happens is this. This is the transcription unit for PDGF. And the skin cell has all the genes, but it's not making this because in the skin cell, this regulatory region is not active. This must have an enhancer that binds to it where? In the megakaryocyte cell in the bone marrow and activates expression in the megakaryocyte. But it's not expressed in other cells. It's not expressed in the skin. But look what happens here in this unfortunate translocation mutation. This gene is a cis-regulatory gene for a gene that's called skin collagen. And collagen is a protein that you probably have heard of before. It's in connective tissue, ligaments, joints, it's in our skin, and it's secreted from the cell, and it forms long polymers that help strengthen the tissue. And so this is normally made in the skin, so there must be a transcription factor that binds to an enhancer that normally activates this gene expression. And so with this translocation, what we get is we get a gene that's this hybrid gene made by accident that joins these two together and is going to stimulate transcription of PDGF in the skin cell, like that. And so we're going to start making PDGF in the skin cell, and what's going to happen is this. Here's a skin cell, and it's all the skin cells in this patient. Oh, well, we'll talk more about that, but it could be all the skin cells, or it could be just the skin cells that are in the tumor. And so here's a skin cell, and it has PDGF receptor. And when PDGF binds, it's going to activate the receptor, and through a signal transduction pathway, it's going to stimulate this cell cycle. Now, in this patient who has the chromosomal translocation that we just described, now PDGF starts to get made by this patient. That's not normally done. It has the gene, but it should not make it, and the PDGF gets released. And even though there's no wound, no injury, we're prepared to respond to the presence of PDGF, and that PDGF doesn't come from platelets, it actually comes from the patient's own cells, and it just creates a stimulation feedback pattern. And in endocrinology, this is called an autocrine response. It's just self-stimulating. One thing that's really terrible about this is that in a cancer patient who has this type of cancer, if the cell floats away, and in cancer biology that's called metastasis, and you've heard that before with that cancer can spread. So metastasis is the cancer can spread. And in this case, if just one of the cells floated away from the skin tumor, and say it floated and then ended up in the lung, it would just keep growing there. So it's a really devastating cancer because just one cell is enough for the patient to have cancer. Okay? All right. So because this is an advanced biology course, I'm going to tell you one more thing, and that is that this maps out here specific patients. And so see here, it'll be the number of cases. So they're doing this where they're looking at a whole bunch of patients and they're mapping the exact breakpoint like that here, and the translocation brings together these two pieces of genes, the skin collagen gene and the PDGF gene. And you could see the skin collagen gene could break at several different points, and then it joins to a pretty specific position on the PDGF gene. And it actually is a little bit different of a translocation than we've talked about so far, because the protein product that gets made is actually partly collagen. So this shows the protein product. So it's partly collagen protein and partly PDGF protein. So in fact, in this specific example, the translocation occurs, I said, cis-regulatory region and transcription unit, and actually the translocation occurs a little bit in the coding region of the two genes. So it's even a more specific translocation than we mentioned before. So I'll just redraw these genes and show you that, when they look in detail, here's the coding region of PDGF, and here's the coding region of skin collagen. And we're used to the idea that when we're designing the gene in a lab, we would just, have the break here, and that's how I drew it before. But I want to get a little bit more specific to this because there's something that in our advanced course we'll appreciate, and it's really a difficult thing when we think of that poor patient, and that is the break points are actually in the coding region. They're not really between the cis-regulatory region, and they're in the coding region. And so what we're going to make is we're going to make a hybrid protein. The actual product is not just PDGF. The actual product is a chimeric protein. Like that? And that's a problem for this reason. So that's what's shown here, is that in all these patients, they show actually what got made was a chimeric protein, like that. A portion of PDGF and a portion of collagen, like that, like we talked about in class, okay? So the reason that happens, and is unfortunate, is because collagen is normally a secreted protein. Where we see collagen, that's in connective tissue, and connective tissue has the collagen outside of the cells in a gel-like material called the extracellular matrix. The PDGF is a cytoplasmic protein. Remember, it got made in the megakaryocytes, and it's just floating around the cytoplasm, and the contents of the platelet is part of the cytoplasm. When we said that, and this just shows a picture of there's, the primary structure of a protein, and we're making this chimeric hybrid protein. And the reason that that is a real concern is because if we just made PDGF in a skin cell, the PDGF would just be floating around inside the cell. It's not going to get released outside the cell, and it actually wouldn't be a problem because the receptor, PDGF receptor out here, has the binding site on the outside of the cell. Remember I said that skin collagen is normally made in the cell and gets secreted. It gets made in the rough endoplasmic reticulum and onto the Golgi and onto the cell membrane, and the skin collagen gets released by exocytosis. And it's actually the first part of the skin collagen protein that has a signal sequence that targets it for secretion. And so what we have in our chimeric protein that gets made by the patient is the first part of the protein is skin collagen, and the second part is PDGF. And that's how the hybrid protein gets out of the cell and binds in an autocrine fashion back to the receptor, the PDGF receptor. So it's a very specific translocation that occurs by random chance. It's very rare cancer because the chance of us having a translocation that occurs is very low. The error of like joining the two chromosomes together that got broken, at the same time, and joining them joining them together incorrectly is really rare. And then it would be anywhere along the chromosome that it could occur. It could even be between the two genes, but it has to be within the coding range of those two genes to join the portion of the skin collagen coding region that's going to signal it to be secreted with the business part of the PDGF coding region that's going to bind to the PDGF receptor. So what a tough story, and cancer stories at the molecular level are always really tough, unfortunate stories like that. We're like, darn it, that is so rare and so sad that could happen. Okay? All right. So that's what happens in our patient. And now we're going to go on to the part where we're going to draw the normal signaling pathway so we can see what is happening in both the normal person under a scab and in this cancer patient that has this very specific translocation. Okay? So here's the pathway. It's in our receptor chapter. Here's the PDGF receptor, so we want to include that on our drawings. And it is a receptor tyrosine kinase. It's in the cell membrane, so there's the cell membrane. And these receptors are transmembrane receptors. That means they span, they cross the membrane, and we have an outer portion that's going to bind to the PDGF, normally under a scab, or the chimeric collagen PDGF that's in the skin cancer patient's tumor. And that's going to stimulate the receptor to be activated, and the PDGF receptor is a protein kinase. And in this case, what these receptor tyrosine kinases do is they phosphorylate themselves, and we'll learn a little more about that. So they're going to autophosphorylate, and that's going to activate an internal pathway where proteins are going to bind to that, and they're going to activate other proteins. So when PDGF is present, this protein binds to it and activates RAS. And we've talked a little bit about RAS. RAS is in a growth signaling pathway, and it's normally in that. So RAS is active under a scab. So there's a scab, and in the skin cells underneath it, RAS has been activated because the PDGF receptor is activated, because the PDGF is bound to it, because the PDGF got released from platelets. So see, we just have a series of steps and more that are cause and effect relationships so that we could say all these things are happening under a skin cell. And then the RAS is going to activate a protein called RAF, and RAF is a kinase. Remember, there are hundreds of kinases, and RAF is going to activate MEK by phosphorylating. MEK is a kinase, and MEK is going to activate ERK by phosphorylating it, because MEK and ERK and RAF are all kinases, and then ERK is going to phosphorylate a protein called ELK, and ELK is a transcription factor. So I think I have it in my next slide that, it's going to be a little bit ahead, that, remember, we're talking about transcription factors as a big part of our course, and we have examples, and we first learned about myogenic muscle, FOXO, and then we're learning about the estrogen receptor as examples of how these get added, how they get switched on, and ELK join, this is just a typo, this should be ELK. We're going to add ELK to our list of transcription factors, and this one is comparable to FOXO. FOXO is phosphorylated by kinase, like that. In that case, if FOXO is inactivated, in this case, phosphorylation activates ELK. And there are many more examples of transcription factors that get activated or inactivated by a kinase, by getting phosphorylated. So that's what we're going to especially focus on, because, where did we go? Because in this pathway, ELK is going to activate transcription, and it's going to activate transcription based on activating ELK target genes, so genes that have enhancer that ELK protein binds to when phosphorylated. It's going to activate transcription, and one of the genes, just one for now, that gets activated is called MYC. Okay, so all that is happening under a scab, and unfortunately all that's happening in the woman who has the skin cancer, and that is PDGF is bound and activating PDGF receptor, which autophosphorylates, and causes it to bind to this protein, which activates RAS, which activates RAF, and then that RAF, MEK, ERK are all protein kinases. They're amplifying or increasing the signal, and we'll learn more about that in the receptor chapter. Ultimately, a major story is they're going to activate ELK transcription factor by phosphorylation, and ELK will bind to genes like MYC and activate MYC. MYC, and I think I have an example of this, so these are MYC target genes, and there are a bunch of them, and for us, our story in the skin is that it's going to include cyclin genes, and cyclin genes make cyclin protein, and cyclin protein is necessary in the cell cycle for start and to allow the cells to go through start. So I'm just going to scroll back to here and say, back to this image of the cell cycle, is these cells are waiting here, and they're waiting here because they don't have enough cyclin protein. And so the PDGF is going to stimulate the cells to heal by increasing the amount of cyclin and increasing the rate, the frequency that they'll go through start. So it's not about how fast the cells go through start, it's how often they get to start the cell cycle. And they roll around and they go through mitosis, and we see an increase in mitosis because that start is occurring more often, so we just have more cells cycling. Like that. So the patient with skin cancer has a very high mitotic index. We have a high mitotic index under our skin cells, like that. Great, it all fits together sensibly. Okay? All right, so that's what we're looking at. That's what we draw, because as molecular biologists, this is a story we're really interested in. One of 58 receptor tyrosine kinases in our body, the PDGF receptor gets activated by PDGF and stimulates the cell cycle through this pathway. We'll learn more about RAS and so forth. And we want to name these and draw these out in the pathway. And you might have to make multiple drawings. Maybe the first drawing would be PDGF driving the cell. Second drawing showing how it activates the genes. Third drawing maybe showing that the focusing in on the MYC gene, and showing, well, phosphorylated ELK binds there, so we're going to get MYC, and MYC is going to stimulate expression of cyclin, and cyclins are going to help stimulate the cell cycle to move through start with that. So that's what we're at on that. So here's the PDGF receptor, and that's really the main protein we think of. We'll see that RAS is something we talk a lot about, but it's used in a lot of different pathways, and so when we think about PDGF and wound healing, we think especially about that PDGF receptor. That's kind of the story. Is that's the the one that gets activated and everything happens downstream of that. So PDGF is a growth factor. It stimulates the cell cycle, and so it's got a great name. PDGF growth. PDGF is platelet-derived growth factor. The receptor is a receptor tyrosine kinase, so it's a good example of that. We'll learn about a couple of other examples. And the signal transduction pathway happens, and we have famously RAS, and that RAF, MEC, ERK are all together called MAP kinases, because they're all doing, they're kind of doing the same thing, the signaling pathway cascade to amplify the signal. Then we have the example of ELK, transcription factor, activating gene expression. So the cellular response is going to include activating gene expression of MYC that turns around and activates cyclin and other target genes. Great. Okay, so moving on from there, just back to this, that's a specific example of one type of receptor, PDGF receptor, and ligand, PDGF, and activating a cascade of signal transduction that includes a lot of phosphorylation and ultimately a cellular response. And in this case, it's to stimulate the cell cycle, grow and divide, like that. Okay? And I think that's it for this video. Awesome.